Hi, everyone. My name is Heba Ismail. I'm an associate professor of pediatrics and pediatric endocrinologist here at Indiana University. I'm also a physician scientist, and I study different aspects of type 1 diabetes. But I'm excited today to share some of the work that we've done looking at the gut microbiome and metabolome in type 1 diabetes, and particularly in obesity and type 1 diabetes. So first, let's talk about obesity and type 1 diabetes. There's been, unfortunately, several studies, whether in adults or pediatrics, looking at the prevalence of overweight and obesity in T1D, and it appears to be really high. But what's most concerning is that it is highest up to 46% in certain underrepresented groups and minorities like Hispanics and Latinos. We know from epidemiological studies, such as the TrialNet Pathway to Prevention study, that relatives who are generally at risk for developing T1D, if they have a higher BMI or obesity, they tend to progress faster to stage 3 or clinical T1D. There are clear differences in their residual endogenous insulin secretion. And obviously, they have reduced insulin sensitivity, which makes their management challenging in clinic once they are diagnosed with clinical T1D. But again, what's most concerning is their higher risk for long-term cardiovascular complications, and even at a younger age. So first, what do we know about obesity and the gut microbiome? So there's been several cohort studies looking at either adults or adolescents with obesity and comparing their gut microbiome composition to their lean counterparts without diabetes. And it appears that they have this unique signature that even they don't share with household members or relatives or their twin pairs. Um, there's been also some preclinical studies. So the question always comes up, is it really causing obesity or how is it contributing? And there's been these preclinical studies looking at transplanting fecal microbiome, transplants from individuals with obesity into animal models and inducing obesity that way. There's also several clinical trials, and here this is a meta-analysis of these trials whereby they used lifestyle interventions. But in these trials, they also collected stool samples from the individuals who participated. And you can appreciate here the responders, the green dots, and the partial responders in blue, whether in the figure to the left or to the right, they tended to cluster together um, in terms of their microbiome signature and how that predicted response to the lifestyle interventions compared to the non-responders in red. There's also this meta-analysis, although of smaller studies, that looked at fecal microbiome transplants from healthy donors who were lean into individuals with obesity. And they were able to induce weight changes or weight loss in those individuals. Although you can appreciate there was some variability there in the degree of response. And now we're learning more about the gut microbiome and how they function, and their function and the metabolites that they produce are really what interact with the host metabolic pathways and therefore affect health. So this is a depiction of the gut lumen, and there are certain bacteria known as fermenters that will ferment fiber, dietary fiber, to produce these SCFAs, which stands for short chain fatty acids, which separately from this talk have been shown to have anti-inflammatory effects and immune modulatory effects. But in the context of obesity here, they induce incretin hormone production, so GLP-1 and PYY, which then stimulate the satiety center in the brain. And then they also induce browning of the adipose tissue. And then the other metabolites of interest here are secondary bile acids. So they're also produced by the gut microbiome. Um, and they also induce incretin hormone production with all its effects, but have anti-inflammatory effects and can improve insulin sensitivity through the interaction with the farcinoid X receptor. What do we know about type 1 diabetes and the gut microbiome? Well, we know from several large cohort studies in different populations, different ethnicities, that it appears to be the gut microbiome composition changes happen even right before seroconversion to antibody positivity. So there's some correlation there that appears to be of interest. We also know from the TEDdy study that followed those genetically at risk and collected stool samples from them in the first 5 years of life and compared their composition to the healthy controls, that even though there weren't any differences in the composition, there were more genes related to fermentation and the biosynthesis of these SCFAs in the healthy controls compared to those who developed islet antibodies. Also from the TEDdy study, now speaking to the role of the virome, it appears to be that there was an association also between prolonged enteroviral shedding and development of autoimmunity. But then when looking at T1D and obesity and the gut microbiome, there wasn't anything in the literature to describe what their gut microbiome and metabolome looked like. So we set out to better understand that and try to identify the role that the gut microbiome might be playing in obesity and T1D. So obviously, the first step was to just compare cross-sectionally the gut microbiome composition in individuals with T1D who were lean. Here, 27 individuals, um, and those with obesity. Here we have samples from 21 individuals, and here the BMI is defined according to the CDC percentile charts. Average age for this population overall was about 15 years, average diabetes duration about 5 years. The percent female was a bit lower in the individuals with obesity, but not significantly so. And we also accounted for that in our analysis. They did have advanced puberty, Tanner stage 4 or higher, and then the average glycemic control is typical for that age group, so 7.5 to 8%. Total daily insulin dose was significantly higher in those with obesity, but after calculating per kilogram body weight per day, there were no significant differences. So first, we looked at the alpha diversity of the samples. So that usually is to look at the richness of the samples, the variety within the samples. And here you'll always see the lean group in some shade of blue in the results that I'll share, and then the group with obesity with some shade of orange. And we saw no differences in the within sample diversity measures between the groups. Then we looked at something called beta diversity, so trying to identify the types that clustered on this principal component analysis by BMI group at the family level, and you can appreciate some clustering here in the group with obesity for Prevotella, and then Bacteroideaceae for the lean group. We then looked at certain abundance ratios. So the first one to the left here, firmicutes to bacteroidetes ratio, that is typically described as lower in people with T1D. And actually, in individuals with obesity who don't have diabetes, it tends to be higher. And here we saw the reverse for those with T1D who were lean, although there wasn't a significant difference when comparing to the group with obesity and T1D. And then to the right, Prevotella to bacteroides ratio is a ratio that's been looked at and described as significantly higher in people with obesity without diabetes, and appears to be related to activation of certain reward centers in the brain in studies that did imaging of the brain. And here we saw a significantly higher ratio in individuals with T1D and obesity, so consistent with the other literature in obesity. We then wanted to identify taxa that best discriminated the BMI group. So for that, first we did the linear discriminant analysis, effect size, to look at the microbial community relative abundance. And here to the left, you can see that there were specific taxa that did appear to be of interest here. Prevotella copri is one that's been described to be associated with obesity, and some others that have been described to be associated with disease state. And then in the lean group, Bacteroides, several of them appeared to be of interest here. We then looked at a subset of 16 individuals in each BMI group, so total of 32, who were matched for age, sex, and race. And again, we were able to identify certain taxa that discriminated both BMI groups. We also I do want to go back to this slide. We did repeat this analysis using Muslin-2, which is a bit more rigorous, just to again validate this analysis, and we saw the same differences. Next, we looked at the functional pathway differences. So in the unmatched samples, we saw this upregulation of branched chain amino acid biosynthesis in the group with obesity. And typically in the literature, branched chain amino acid levels that are higher are associated with insulin resistance and obesity. And then in the lean group, there was upregulation of tyrosine metabolism and tryptophan metabolism, more related to energy balance. And then the other one that was of interest was upregulation of secondary bile acid biosynthesis. And if you recall, high levels are associated with increased incretin hormone production and improved insulin sensitivity. Then in the matched samples, we saw the same findings, but also what appeared that was new this time was upregulation of branched chain amino acid degradation, thus suggesting or inferring that perhaps the branched chain, branched chain amino acid levels were lower in the lean group. Next, we looked at stool short-chain fatty acid levels by BMI group. And interestingly and surprisingly, their stool SCFAs were significantly higher compared to in the obese group compared to the lean group. And that was all three main SCFAs, so acetic acid, butyric acid, and propionic acid. And we tried to correlate that with the diet history that we were able to collect. And we saw no differences or associations with different dietary components, including fiber. With that said, obviously, there's always the recall bias with diet history that could be affecting this. So then we were left scratching our heads and like what we thought SCFAs were good for you. So we went back and started looking at the literature, and actually we had missed it. So there were several studies there showing that higher BMI is associated with higher stool SCFAs. So this study showing the highest tertile in blue had the highest stool butyrate level. And then in this study, looking at lean and overweight individuals this time, not obesity, the stool SCFAs, again, all three main ones were higher in the group that was overweight, especially after adjusting for age and available carbohydrates. Then this study also compared the stool SCFAs and looked at their levels and how they associated with different metabolites, but also the incretin hormones and in a subset of individuals that underwent clamp studies looking at insulin sensitivity measures. And there were no associations here with the fecal levels of these SCFAs. But instead, when they looked at circulating levels, there were several significant associations here after adjustments. So, for example, here looking at GLP-1 right in the middle of the table, there were significant associations with all the circulating SCFA levels. And then similarly for insulin sensitivity measures, there were significant associations there. And so the postulation became that perhaps what's happening in the in the gut of individuals with obesity is that the gut microbiome there has some increased energy harvest whereby it's either holding on to the SCFAs for themselves, not sharing them, or the gut is not absorbing, and thereby the host, the individual or the human in this case, is not seeing those circulating SCFAs. So then the next logical step was for us to check the circulating SCFAs. And we saw the opposite here. So in fact, in the lean individuals, there were higher acidic, butyric, and propionic acid, although only significantly higher for acidic acid. And then the same for secondary bile acids. So Gutka and Tutka are the ones that have been described to be associated with increased insulin sensitivity. And these were again higher in the lean group. Now, note here, this is preliminary data. These metabolites have not been, the levels have not been normalized yet. So this is very early, but it goes with that notion that it's really the circulating levels that are important in this situation. So before jumping on to therapeutics, I just want to say that obviously that was early data. We had to see what if there are differences to start with cross-sectionally, and it would be very important to validate this in a larger cohort. So that's what we're doing right now. We're also we're planning to collect samples longitudinally from individuals but that will take time. And then we just got a grant also to take some of these stool samples from the individuals with obesity and T1D to transplant them into germ-free mice to see if we can also induce obesity and perhaps insulitis. So there's more to be done. But very quickly, going through therapeutics, just so we don't go over the allotted time. But here, there's been several medications now that we're learning are starting to learn that they work through the gut microbiome, at least partially, and one of them is metformin. So metformin has been shown in several studies to actually increase SCFA production and secondary bile acid production and thereby increase insulin production and also insulin sensitivity. So it's a very interesting medication that way. And here we had serum samples from individuals who participated in a 26-week clinical trial where they received either metformin, they were randomized to metformin, or placebo for 26 weeks. And this is actually a T1D exchange trial led by Ingrid Libman and Kristen Nadeau. And we found here in the heat map, you can appreciate the metformin group is in blue, and then placebo in red post-treatment. There were significant differences, and these are the top 34 significant features. But then of interest too were three that were of microbial origin. So, for example, allantoic acid, that's been described to be typically higher in individuals with obesity and insulin resistance. And here it was significantly lower in the metformin arm, also 5-hydroxyindole and 3-indoxyl sulfate. These tend to be lower levels are associated with either gut dysbiosis or insulin resistance and obesity. And here we saw significantly higher levels of these metabolites. Now, more work needs to be done. Obviously, we need to look more closely at SCFAs and secondary bile acids and also look at changes from baseline. But this was preliminary data that I wanted to share. And this is in collaboration with Dr. Tim Garrett at the University of Florida. Another treatment or therapeutic that's of interest is high amylose maize starch. That's been esterified, whereby acetic acid and butyric acid, so those two SCFAs have been added to this fiber that's an indigestible fiber. And here, this is a small pilot where we gave newly diagnosed adolescents with T1D this HAMS AB or high amylose maize starch AB that's been acidylated and butyrylated for 4 weeks. And we saw a significant change in the relative abundance of bifidobacteria in general or Bifidobacterium longum. Again, this is a pilot, so small sample size and some small changes here, but it was interesting. And then the other thing that we saw were metabolite changes in response to the HAMS AB. So baseline before the HAMS AB is that row at the top there in green, and then red is post-treatment 4 weeks after. And you can appreciate again in this heat map the changes. These are the top 50 features using untargeted metabolomics analysis, again in collaboration with Dr. Tim Garrett at the University of Florida. The ones that were of interest in this analysis were increased hippurate and tryptophan levels. So hippurate is a purely microbial metabolite that's associated with increased gut bacterial diversity and actually improved glycemia. And then tryptophan is partially produced by the gut microbiome, but it's anti-inflammatory and regulates energy homeostasis, obviously. And then the other two were increased L-glutamate and dihydroxyquinoline. And these are intermediate metabolites also produced by the gut microbiome and are associated with increased diversity and improved glycemia as well. So this is interesting. Now, also note here that when we looked at glycemic changes, we actually did not see significant changes, but this may be just related to, and it was measured by A1C, so it may be just because they received this for 4 weeks. Although I will say anecdotally in clinic, for those individuals that we follow in our clinic, they continue to have amazing glycemic control. So that wraps it up. So I'd like to end by thanking funding agencies and the grants that supported all of this work, my mentors especially, and collaborators, obviously within IU and outside. and then the study team that helped us get these samples, and obviously participants. So yeah, if you have any questions after you watch this video, please reach out either via email or LinkedIn. Thank you so much.